Jane specifically said I'm not allowed to talk about IMAS or VIMA because it's actually fantastic products in Africa, but Professor Jennifer Thompson was going to speak about it, and it's specifically like water-efficient maize for Africa, which is a fantastic product, and then also IMAS, which is improved maize for African soils, very poor in certain nutrients. So my talk is going to be specifically then on nutritional quality enhancement and then all the things that we actually do in tobacco plants. I always wanted to use tobacco for something different than smoking. Okay, so I'm first going to touch on the African Elite Maize line. It was interesting. What I want to portray here is, I'm going to touch on nutritional quality enhancements, and specifically the European partnership almost with South Africa and how we actually can bring a product to the market. But this was just a sort of something that happened that wasn't particularly planned. There was no particular project for it. But we screened something like 30 different maize lines just to see their responsiveness in tissue culture and if you can transform it. And we came up with this public line M37W, published in 2001, and we were sort of introducing all kind of traits, and then eventually myself and Melanie Vivier, we went to Department of Science and Technology, and we wanted to put in all kinds of carotenoids into the maize and into the sorghum. But it's not a lack of interest, I think, of Department of Science and Technology necessarily, but just a small panel that handled it, and they turned it down, and they decided, nope, not carotenoids into this maize and sorghum. And then 3 years later, Paul Christou took it up, and he actually took M37W, South African maize line, and he introduced all these carotenoids into it. And he had a whole library of phenotypes that they actually developed. And this is just a painting that Therese Capel then made, and they work in collaboration. And it's all the different carotenoids that's introduced into the maize, and you get all kinds of colors. And the most interesting one, unfortunately, I cannot speak about because they're going to patent it, but it is a fascinating approach also. Then Africa Bio, they are actually a weekly newsletter almost, or 2 weekly at least, and they portray all the different challenges in Africa, bringing biotechnology products to the market, the policies, and addressing basically everything concerning biotechnology and the releases in Africa. And then maybe what I just want to also mention is the potential for Africa. My colleague, Dr. Luc Methlo, then took it up, and he was a colleague of Paul Christou previously, so they came to visit, and we're going to see if those kind of products can actually be released in Africa. Now, of course, maize is contained by just detasseling, so GM maize is very welcome in Africa. When we talk about sorghum and palmate, it's not as welcome. It's because it's indigenous to Africa and it's open pollinating species, so it actually can just spread. So I think the majority of my scientific career was actually in genetically modified organisms, but I dropped it and went to tobacco purely because it is so difficult. The regulatory pathway is so challenging to bring these products really to Africa for the benefit of Africa. And then, of course, also Florence van Bugu, very powerful woman in Africa. She's based in Kenya, from Africa Harvest, CEO of Africa Harvest, and tremendous person that really wants to bring the benefits of biotechnology to her people. I mean, she even had in her backyard bananas planted so she can understand the market of how to bring it to the market. A fascinating person. But then she's leading the Africa Biofortified Sorghum, and majorly with Pioneer. We had a part in it, but Pioneer, I think, is the main partner because they have the technologies that could benefit and elevating. Now, initially, they wanted to actually look at all the different micronutrients as portrayed there, iron, zinc, lysine, all of them, but eventually started to focus only on provitamin A because that is really the deficiency in African crops that's really lacking. And of course, micronutrient deficiency, or called the hidden hunger, is almost only obvious when it's too late. Of course, your children becomes blind and they die. But then also, apart from genetic modifications, there's also alternatives, many alternatives. So first of all, the regulatory pathway is very challenging. Then there's fortification and supplementation that can take place. And then, of course, also in 2001, in May, 50 partners came together and they want to bring provitamin A to Africa in orange sweet potato. So, I mean, it is available if you can convince communities to actually take it into their diet, on a daily basis. And then, of course, there's also Harvest Plus, that's non-GMO, but would also breed for sort of elevated levels of different nutritional targets. Then, of course, there's also mutational breeding. And yes, their pathway to the market is so simple. So I think Dr. Luke Mecklenburg and Zotwa Mbambu, they're also involved with this, and they actually elevated lysine levels purely by mutation. And then the drought initiatives, I maybe just want to touch on it because it was spoken about quite a bit. We also have some project which is really marginalized. It's not really part of our bigger pool of projects, but we're actually looking at sorghum land traces and identified 4 out of a pool of 45 that shows sort of drought tolerance or resilience to droughts that's sort of similar to what the drought-tolerant elite breeding lines would be. And we're currently actually evaluating and looking at it to actually see, is there a specific genetic makeup in this land races that makes it more tolerant? And then what I wanted to mention about Nomad, I don't know how many of you know it, there was talk about ICON, the FP7 program. If I say ICON, I see ICON Genetics. And Yuri Gleba was actually the CEO of ICON Genetics, and using deconstructed viral vectors to express all kinds of proteins in the plants. Now, Yuri is actually, he just thinks outside the box. So, I mean, that was actually sold to Bayer, and they driving biopharmaceuticals. And now Nomad is actually focusing on biomaterials and then also on agronomic traits. So what he does with this Nomad, and I think he got it from the desert, you know, with the sandstorms, so you basically have an abrasive type of thing, and then also overexpression of certain particular proteins that would protect the crop. So say, for example, there's drought this year, you just spray it, you know, the protein is expressed, it's not GMO, and actually protects the plants, and, you know, it's drought resistant. And the next year there's floods, you're protected against floods. So that is what Nomad is all about. And then Cameraplasty, wasn't mentioned, but that's something that actually in the latest proceedings for National Academy of Sciences was also thought of as potential, you know, target genes, agronomic traits, to actually change it just by mutation, but it's a targeted approach. Now, previously it's been used, it's in science for a couple of years, but mostly used in cancer therapy. Okay, then I just really want to touch on microbial expression systems. That's also part of CSIR. And we have, of course, the whole pipeline. I think it's one of management's best decisions ever. They put all the people together, that is microbial, plant expression, and yeast expression systems, all in one lab. And all the kind of interaction that takes place there. So I sort of want to represent some of the activities that they're doing. So basically, from proof of concept right through to commercialization, that's basically what they would be doing. And then we have already spun out companies, and then I want to focus specifically on Optimus Bio, which is bacillus specific for probiotics and also water remediation. And then resin is basically also a resin, say for example for purification of yeast-tagged proteins, but it's just quite powerful kind of resin that they developed, and I think AAC Amersham has actually purchased it, and they will actually be selling it. It's just not scalable as some of the other resins. So that's something that they still have to work on. And then BIDC is also part of Biosciences, and it actually is Biomanufacturing Industry Development Center, and they would incubate small and medium-sized ideas that they want to develop actually to market-ready products. So they would translate all kinds of ideas, technologies into market-ready products. So that's really what BIDC is, and they actually had quite an input from government through the job funds, had 90 million rands to actually incubate small and medium-sized businesses. Then I think what I'm doing the last 4 or 5 years, and I know there's many people that's far more experienced than us, but it's something that's absolutely fascinating, and that is using deconstructed viral vectors to make all kinds of proteins, biopharmaceuticals, vaccines, antibodies, into the plants. And specifically, we're using tobacco, but I mean, if you know the world, what they're doing, they use anything from duckweed to carrot cells to even strawberries. And you can literally just pellet it, make it into a tablet, and your interferon is there, and it's amazing, and you can control the dosage, of course. Okay, so this was really the original, when we started off, Dr. Rachel Chikwamba, and she's actually our director at present. But she started off with making a cocktail of 2 rabies antibodies. Now, the sequences was obtained from World Health Organization, so right through the project, they were actually almost partners in assessing the product. So first of all, they've actually confirmed that it's as equal as the current what is a gold standard in rabies antibodies, and that was made in the plants. And it's also something like 500 milligram per kilogram, so it's quite a high expression of these antibodies. So of course it was validated in vitro and in vitro for efficacy, and also been once again validated with the World Health Organization laboratories. We also looked at neutralization patterns against 30 rabies strains, was very effective. It was made on a CGMP, and that is certified good manufacturing practices at KBP, which is our global partners, and they are Kentucky Bioprocessing. And then the glycoprofiling was done in our labs also by Dr. Stoikev, and then also once again validated in Austria. And eventually we are at the point where we have a preclinical trial, and we can bring it potentially to that. And I think then MAP was really in the news lately, and that was producing ZMAP, which is antibodies against the Ebola. And there's 3 murine antibodies that actually is in the cocktail, and we probably will negotiate to actually produce it then in our laboratories. But I think something that we do lack, and the government is looking into, is that we don't have a CGMP facility to produce the products that we're developing. So that is something that they're very interested in, because even for a pandemic type of response, they're interested in that type of activity. Then we also have a license agreement to use the TMVHA, and that's basically tobacco mosaic virus, but it's infectious tobacco mosaic virus, and it actually carries the human influenza antigen, and then incredibly effective. And then it was published recently where it actually, it will give a 50% sort of protection, as is, just the product as is, and then 100% protection with the adjuvant. So it's a very, very powerful kind of... Then I think something that I'm very passionate about, something that we're doing currently in our labs, is creating virus-like particles. Now, I've said the tobacco mosaic virus that we get from KBP is actually infectious. You can imagine if one of them escapes and actually wipe out my growth room facility. So I definitely don't want to go that route. So what we want to do is using virus-like particles. Now, this is a fascinating field. I can walk, yes. Here you can see this is actually how Mark Doust actually portrays it. Here is a real virus, or there would be a real virus with a viral genome. Virus-like particles will be completely free of the viral genome, which means it cannot replicate and it cannot infect. So it's a powerful system. So what you have your 4 capsid proteins, you bring them together, you have your core-like particles, you have your virus-like particles. Very powerful presentation platform. You can use it as is against specific diseases, or you can actually conjugate an antigen. So this is something that we're working on, but we actually have a particular product that we want to patent at the moment, so I cannot speak about that, but hopefully soon. And then we do have partners like Honest Word Biological Products, that's specifically veterinary, and most of our products at the moment is veterinary products, purely because it's easier to bring it to the market. Eventually we can penetrate the human biologics kind of market. Then I think the leaders in the field is Fraunhofer, USA, specifically USA, in developing virus-like particles, and then Medicago, and that's specifically Marc-André Dalst, and he developed, rabies virus-like particles, and also human influenza virus-like particles, and it is a very potent vaccine that can be used. It's completely self-adjuvanting. Then just to also mention our colleagues in the south, they're not really colleagues, they're maybe our competitors, but they've already brought quite a number of virus-like particles also to the market. Of course, our target products is very different, and that is Professor Ed Rubitsky's group. Now, at University of Cape Town, they're actually developing it over a couple of years, 15 years, they're into this type of work, but only the last year were they actually registered as Biofarming Research Unit at Cape Town University. And what they've created, and all the red ones is already patented, so it's all vaccines for human health. And it's the human papilloma virus-like particles that's been used, influenza, but specifically the H5 polypeptide, then rotavirus in collaboration with Doust, which is from Medicago, the leaders in the field, and then also the HIV gag virus-like particle. The other 2 is really veterinary vaccines that they developed, and a subunit vaccine, and not necessarily virus-like particles. So that's really the things that I wanted to bring to the table, and that's just some of my colleagues that's working on these projects.